So thank you for putting together this very informative conference. We are glad to be here. And today we'll discuss our program, so which is SLS005 trehalose for ALS. And one of the common theme, as you saw earlier from the previous presentation, that autophagy is a it was recently awarded a Nobel Prize in 2016, where genes identified that were responsible for autophagy. And since then, there has been a lot more research being conducted in exploring this, and that we are part of that. So we are exploring autophagy as a common pathway in ALS. What is unique and good aspect of this exploration is that this will be for the treatment of both familial as well as sporadic ALS in the prominent mutation types. Just a brief history of the role of autophagy in ALS. So practically speaking, if we focus on the phenotype of the disease, it manifests itself with the progressive muscle paralysis, accompanied by the degeneration of the motor neurons in the spinal cord as well as in the cortex brainstem. So that's the phenotype of the disease, and followed by the genotype. What you see is the accumulation of abnormal protein inclusions. The four prominent ones, and of course more are being discovered as we speak, is this the SOD1, TDP43, FUS, as well as the chromosome 9 open reading frame 72. These abnormal protein inclusions have been identified as a pathological hallmark of ALS. And underlying theme is, so the question is the dysregulation of autophagy that has been implicated in the broad spectrum of the ALS patients. Is that a definitive cause and effect in the pathology of the disease or not? Some of the earlier findings are very encouraging. And before we get into that, just to explore what the disaccharide trehalose is. It's interesting that the humans don't produce trehalose, but we have an enzyme, trehalase, that specific purpose is to hydrolyze trehalose. So it metabolizes it, and we have it, we humans, mammals, and it occurs in other mammals as well, in the gut, kidney, and liver, and a small extent into the CNS as well. However, we don't produce any trehalose. So it must be some evolutionary rudiments. And so trehalose, in order to be an effective therapy, must be given through an IV infusion to bypass this trehalase enzyme in the gut. The other unique aspect, of course, is trehalose being a small enzyme, so biodistribution in the CNS as well. But it's also in the muscles, and as we earlier have seen throughout this conference, that the therapy has to penetrate the muscles as well, because the muscles atrophy or paralysis is part of the ALS pathology. So we need the therapies to be distributed both in the brain and the muscles, and trehalose does that. In terms of the mechanistically, the simplest way to put it is, and there's multiple studies done in this, is trehalose is a master manipulator of autophagy. It enhances autophagy, and primarily, if you look at the third bar on the right, it basically activates the transcription network by activating the transcription factor EB that'll translocate from the cytoplasm into the nucleus and activate both the lysosomal pathways and the autophagy genes, through which these mutant protein aggregates of TDP43 or SOD1 or similar pathological accumulates can be cleared out. Recently, another publication has come out from INSERM in France, where they have implicated specifically for TDP-43 aggregates that within the neurons, one of the 2 or 3 mechanisms that exist to clear out the TDP-43 aggregates, either through activating autophagy or through activating the UPS, the proteasome systems. And what we have seen, there's plenty of evidence that trehalose activates both of those mechanistically. So mechanistically, we are aligned. In terms of then preclinical findings, what we found was that the effect of trehalose is very similar to addressing both the phenotype and the genotype of the disease. So in this model, it's a SOD1 model. We decrease the aggregates, as well as the preserving of ventral horn mirror neurons, where the ALS gets implicated initially, is also shown, and the induction of autophagy is in place. And one has to be careful in one of these preclinical earlier studies that you will see that the trehalose has to be administered not just orally, which is 98% of these preclinical studies did, but the IP, the intraperitoneally as well, because it turns out even mice have trehalase enzyme in their gut. So the oral way of administering trehalose is not the most ideal, and yet you see the good efficacy in these models. This one is the neuron cell line, and sure enough, you see the activation of the transcription factor EB that I elucidated earlier, and increased clearance of the TDP-43 aggregates. And this is another molecule, another model where we show not only reduced aggregation of SOD1, but also P62 is the well-known biomarker of autophagy. So you see that, and we are preserving the muscle strength, which is important pathology of the disease, and improving the autophagy flux. And in the next model, what you will see is, to the extent some of the pathology is outside the neuronal cells and more in glial cells, you also see the inhibition of microgliosis and astrogliosis upon treatment with trehalose. So preclinical data is compelling, addressing both the genotype and the phenotypes of the disease. And so far in humans, we have utilized trehalose in other CAG-type diseases. And that CAG-CAG is standing for the glutamine codon. So there are other diseases similar to ALS where you have, due to gene mutations, extra repeated polyglutamine. And we have seen pretty decent efficacy in those diseases as well. We have some earlier phase 2 data in spinocerebellar ataxia, which is also a polyglutamine repeat disease, where we have shown the improvement of the patients from the baseline over the 6 months. So these findings make us a compelling argument to do a registrational study through the Healy platform. We have the regimen E, the 160 patients study. It'll start enrolling, dosing patients next month, we anticipate. And in terms of the placebo versus the drug, it's a 3-to-1. 3 patients will get the drug per 1 patient of placebo. So that's the regimen that we are using, and more details of the inclusion-exclusion criteria can be easily obtained from the Healy website itself, as well as people can reach out to us directly. I'll stop here and take some questions.
Thank you very much for a great presentation, and congratulations on advancing into the clinical trial through the Healy platform. A first question from me is there a rationale for also testing trehalose in lysosomal storage diseases? Is that something that you've looked at? And is that more direct than certainly advancing in ALS is what I'm most interested in, but I'm curious about that as well.
And the answer is yes. The lysosomal storage diseases, we are looking into those as well. And the complexity comes there, unlike ALS, where there are very well-defined endpoints, which is the ALS-FRS scale, the functional rating scale. The lysosomal storage diseases, because of they're ultra-orphan, you don't have those endpoints. So we are in the process of, in conjunction with the regulatory agencies such as FDA, we will define those endpoints and then pursue them. But the answer is yes, we are pursuing that as well.
Thank you. I have a question here. Somebody asking if sublingual route of administration is an option. Is there uptake?
And the answer is probably not. What we do know that most of it, very little of it will go into the CNS, and we are thinking less than 0.5%. And as a result, you will have to keep the sublingual or oral doses way high, which will run into trouble in GI side effects. And there is a high likelihood that FDA will not let you do those studies. So the IV is one of the ways. But however, having said that, we are working on other formulation that will bypass weekly infusion. For the Healy platform study, we have made it available to certain patients, home infusions on the days when the vital scales are not being determined. So this way they don't have it. But we are working on a formulation that'll be less frequent than weekly infusion, and and we will unveil that data when it becomes available in the coming months.
And we have two more questions.
Sure.
We have time for. Is development of an analog that wouldn't be targeted by trehalase an option? Could the molecule be modified?
And the answer is you will leave some efficacy on the table, but it's theoretically possible. And we are looking at that as well. But you're leaving some efficacy. The way the trehalose works, it's like a non-mTOR pathways of activating autophagy. It's very unique. So the theoretically possible, but I think, one of the other intriguing ways that we can do it, instead of supplying exogenous trehalose, one can think of producing endogenous trehalose. And those are the kind of formulations we are working on.
And I think you partially answered this during your presentation, but I'll present it again. Why not intrathecal delivery if central nervous system access is important?
The amount of trehalose involved is so large that it'll not, this makes it very, feasible and accessible, and does not require any, invasive way of delivering. Keep this in mind. The only known metabolites of trehalose are glucose molecules. So the side effect profile, we have had patients who have been on this drug for 2 years, is very benign. And so adding that intrathecal component and the amount of trehalose involved is so large is not a practical solution.
Well, thank you very much again for the great presentation. We'll move on.